Okay, good afternoon, everyone. Welcome back. So now we're back in the virus reproduction cycle. We've gone through RNA synthesis of RNA viruses. Today I want to talk about transcription and RNA processing. This is an event that happens only in cells infected with DNA viruses and one family of RNA viruses, as we'll see. So this is what I mean by transcription, the big red arrow. That's what transcription is. It is the synthesis of mRNA from double-stranded DNA. And remember, you can only transcribe a fully double-stranded DNA template. That's the only template that is competent for transcription. So that is transcription. It is not protein synthesis. I know in past years, some individuals who took this course didn't know the difference between transcription and translation. And you need to know that because I throw the terms around loosely, assuming you know it. So this is what transcription is, the synthesis of mRNA. Translation, of course, is the synthesis of proteins. So which viruses do this? All the ones in red here that have double-stranded DNA in their reproduction cycles. That includes the double-stranded DNA viruses, adenoviruses, herpesviruses, polyoma and papillomaviruses. Those are the main double-stranded DNA viruses we talk about in this course. There are many, many others, of course, but these are our models. So they have double-stranded DNA that can be directly transcribed. Then we're going to talk about the parvoviruses with single-stranded DNA genomes. Those DNAs cannot be transcribed. They have to be made double-stranded first. These are the concepts I love, right? Like double-stranded RNA can't be translated. And you can bet that there will be questions on the next exam about them. So get familiar with that fact that double-stranded DNA is the only competent template for transcription. And then the gapped double-stranded DNA molecules, hepatitis B virus, these are not competent for transcription. They have to be repaired, as we'll see. We won't talk about these viruses much in this session, but we will talk about them when we talk about reverse transcription. So those are the three classes of DNA viruses that we'll talk about today that undergo transcription, the synthesis of mRNA from a DNA template. All DNA viruses, when they infect cells, have to make at least one viral protein before DNA replication occurs. Of course, DNA replication is going to happen to make new virus particles, right? And you will be making proteins to package those DNAs into two particles. But before you can do any DNA synthesis, these viruses have to make at least one protein. Sometimes they have to make many proteins, but at least one. No DNA virus can come in a cell and start replicating its genome without making at least one protein. Even the smallest DNA viruses that are completely dependent on the host for all the enzymes involved in DNA synthesis. And we'll see that today. Now, this is a bit of a conundrum because not all DNAs are ready for transcription, as I just told you. The single-stranded DNA viruses, those genomes are not ready for transcription. The gapped double-stranded DNA viruses are not. In this slide, we're looking at the reproduction cycle of a polyomavirus. These are small DNA virus with circular double-stranded DNA genomes, shown on the right there. Polyoma means many tumors. As we'll see later, that's why they were first found, because they cause tumors in a lot of different animals. So polyomavirus, and the virus we're going to talk about that's a polyomavirus is called SV40, simian virus 40. And this is the reproduction cycle of that. And you'll see that here's the DNA of that virus in the nucleus. It's double-stranded. It's ready for transcription. Doesn't have to be fixed in any way. And so that is an example of viruses whose genomes are ready for transcription. They don't have to be repaired or anything like that. They can go in the nucleus and be transcribed. And that's where they have to go for the most part, in the nucleus, because that's where the transcriptional machinery is. As we'll see in a bit, there are some viruses that do not go into the nucleus. I like to say they avoid the nuclear bureaucracy. It's very busy in messy place, that nucleus. Adenoviruses stay away, they remain in the cytoplasm. And we'll see how they undergo transcription. So here are some of these viral genomes that have to be converted to templates for transcription. I'm hammering on this point over and over again because I think it's really important. If you remember this, it will guide your thinking about the reproduction cycle of these viruses. So here first, Adenoviruses have that weird double-stranded circular DNA genome that has a gap in it. It's got a protein attached to one of the strands, a piece of RNA to another. This is no way ready for transcription, so this has to be repaired. This goes into the nucleus, this DNA, and the cell machinery sees it as damaged and tries to fix it. So when DNA comes in the nucleus, that's a common response. And there are lots of enzymes in the nucleus that recognize damaged DNA and they will fix it. So they remove the RNA, they remove the protein, and they fill in the gap so it's now double-stranded and it can be transcribed. Parvoviruses are also not ready for transcription. They are single-stranded genomes, and they have to be made double-stranded in order to be transcribed. I don't call this genome replication. It's more like a repair pathway. And then, only then when this genome is double-stranded can it be transcribed, and then a viral protein will be made that can allow this genome to replicate. And then for completion, we have to include the retroviruses. Although they have RNA genomes, these are copied into a double-stranded circular DNA by reverse transcriptase, which is the viral enzyme included in the particle. And that then integrates into the cellular DNA, and there it is transcribed. And so again, you could look at this genome as having to be modified before it's a suitable template for transcription, because the RNA itself, as it comes into the particle, is not expressed. It has to be first converted to DNA. We will talk about those viruses in a lecture of their own. So again, which viral genomes do not need conversion? The double-stranded DNA genomes. And they can be circular or linear. Doesn't matter as long as they are fully double-stranded DNA. And today we will go through how each of these kinds of templates undergoes transcription. And next time we'll talk about DNA replication. So again, we're going to talk about parvoviruses, single-stranded DNA, a little bit about Hep B, and the double-stranded DNAs in terms of how these double-stranded templates are transcribed. Much of the information that we know, and I'm going to tell you about today, was figured out using viral systems, because viruses were good tools many, many years ago when it wasn't easy to get an individual cellular gene out and study transcription. Viruses provided those tools. The enzyme that carries out transcription, that is the production of mRNA, is by definition RNA polymerase II. This is the enzyme in our cell that makes messenger RNAs, among other RNAs. And in fact, that's one of 3 RNA polymerases, DNA-dependent RNA polymerases. They make RNA from a DNA template. They all initiate synthesis de novo. No primer is needed for any of these enzymes. And the 3 of them are shown here, Pol1, Pol2, and Pol3. Pol2, which is, again, the enzyme that's going to carry out the synthesis of mRNA. It makes pre-mRNA, as we will see. It doesn't actually make the final product. It makes a precursor. It also makes a number of other important RNAs, both for the cell and for viruses, including primary transcripts from which microRNAs are derived, small nuclear RNAs, and long non-coding RNAs. And we know that, of course, many viruses utilize Pol2 to make their messenger RNAs. You can see every virus that has to do transcription will make mRNA using Pol2. Some viruses have been shown to make primary microRNA precursors using Pol2 as well. This enzyme has been found to replicate the RNA genome of hepatitis delta virus. And we'll talk about that later in the course. It's the only RNA virus that can be copied by a DNA-dependent RNA polymerase. That's pretty weird, but it works. And then these long non-coding RNAs have also been found in Kaposi's sarcoma herpesvirus. Now, the other two polymerases are important. Pol-1 makes precursors to ribosomal RNA, so you wouldn't have ribosomes without Pol-1. We don't know any viral RNAs that are made by Pol-1. Pol-3 makes a variety of smaller cellular RNAs, including precursors to tRNAs, the 5S RNA of ribosomes, ribosomal RNA, and a small nuclear RNA called U6. And you'll see some of these small nuclear RNAs in use today. And some viruses, in fact, produce RNAs using Pol-3. So most of the viruses we will talk about today utilize the cellular Pol-2 to transcribe their genes. There is, as I have just said before, a set of viruses that replicate in the cytoplasm, and that includes pox viruses and some of those giant viruses like viruses. They avoid the nucleus, they stay in the cytoplasm. So they enter cells and set up factories in the cytoplasm, so they have no way to access the RNA polymerase that's in the nucleus. Therefore, they encode their own. So pox viruses and giant viruses encode their own RNA polymerase because they don't go in the nucleus. That's logical, so I think you can remember it that way. The viruses that go in the nucleus use the Pol-2 that's in the nucleus. The viruses that stay in the cytoplasm encode for their own Pol-2 enzyme. All right, so here's an overview of what we're going to talk about today: transcription and RNA processing. So here we have a cell nucleus at the top, and you can see the nuclear membrane, and there are nuclear pores, a few nuclear pores there. So top is nucleus, and the bottom is the cytoplasm. So we start with a double-stranded DNA template with the two colors of blue representing one strand and the other. The promoter, that's the site at which transcription begins, I will usually show as a red arrow. So that's where the enzyme is going to start making mRNA. And here that synthesis is beginning. You can see a short transcript is made. That's the first transcript made. It's got a cap, which we'll look at in some detail today. It's an unusual base at the 5' end of the mRNA. And then we make a complete pre-mRNA. And that's shown here, the second line. And the pre-mRNA differs from the final mRNA in a number of ways. First of all, it frequently has intervening sequences that have to be removed by splicing, which we'll talk about today. And in addition, the 3' end has to be polyadenylated. And only when those things happened, we finally have our mRNA shown at the bottom there with a cap, with the introns removed, poly-A tail, and then that can be exported to the cytoplasm. That's why I say Pol-2 makes pre-mRNA, because after the mRNA is made, other reactions have to occur before it's considered mature. And that mRNA, of course, is exported into the cytosol, where it can be translated to form protein. The export of those messages depends on them being spliced, because splicing deposits proteins on the mRNAs that are recognized by the nuclear export machinery. We'll come back to that later because it's important, because some viral genomes have to be exported without being spliced, without having intervening sequences removed, as you can see in this example. And so some viral mRNAs do not even contain intervening sequences, and others have them, but to make full-length genomes, they have to be retained. And so there have to be mechanisms to get unspliced messages out of the nucleus, which is what we will talk about. All right, so let's start the promoter. Again, all of this was pretty much discovered using viral systems. The promoter is the region of the double-stranded DNA, of course, at which transcription begins, and transcription is highly regulated. It's a perfect point in gene expression where you can regulate it. And you do so by regulating the initiation of transcription, by having lots of sequences around the promoter that can be regulated, and proteins that bind to them whose synthesis can be controlled in turn. And so this is a typical promoter. The promoter of this piece of DNA is on the right. It's shown by the red arrow. And the gene that's being transcribed isn't shown here. So we're looking at the transcriptional control region. So right at the promoter, there are lots of sequences that have been identified in these regions. and there are proteins that bind to them to regulate transcription. So for example, right at the promoter, we have in this yellow sequence, it's called an initiator sequence. It's a specific sequence that specifies accurate starts by the polymerase. And then upstream of that, we have in green the TATA sequence. It's a series of TATAs that have been shown to bind a protein called TF2D. And it's very important for the polymerase to start recognizing and initiating there. And those two sequences together are called the core promoter. You could actually take those by themselves and put them in a plasmid and direct transcription of an mRNA. But we have other regulatory sequences as well. The core promoter is typically 20 to 35 base pairs in length, but you can see you can have 100 to 500 bases upstream, other sequences shown by these various colors that bind other proteins, and they're called regulatory sequences. They can influence the expression of this gene, and that defines the promoter. You can see these local regulatory sequences plus the core promoter, all of that is the promoter. Then even 10,000 bases away, up to 10,000 bases away, we can have other sequences that will influence transcription. Those are the purple sequences there at the very left end of the DNA. These are called distant regulatory sequences because they can be 10,000 bases away. They're enhancers or silencers based on whether they augment transcription or whether they silence it. And we'll see how they work in a moment. And so that's a transcriptional control region. It's a series of very specific sequences that bind specific proteins, and the combination can differ from promoter to promoter depending on when it's needed and at what levels. And all of the promoters in the genes that we have within us are all regulated very carefully so that just enough protein, just enough mRNA is made that specify proteins when they're needed. So you can get exquisite control over gene expression. So here are three examples of viral regulatory sequences at promoters in three different viruses. So first we have the Adenovirus major late promoter. I haven't told you what a major late promoter is yet, but you will see it is a really important promoter that operates late in infection. And that's a diagram of the promoter. The transcription start site is shown there at the right, the red arrow, and the yellow is the initiator sequence. But you can see there upstream a couple of different regions. There's our TATA box in green. And then you have an orange and a green segment which have funny labels, USF1 and CPF1. And all these have weirdest names because they're all named by different people as they were discovered over the year. For example, this one stands for upstream factor. It's informative, isn't it? But what it is, it's a very specific sequence that binds one protein that's needed for transcription. So this particular promoter of adenovirus has evolved to have a certain combination of protein binding sites because it needs to be regulated in a specific way. Down next is the SV40 early promoter. Promoter start site is at the right. You can see there are a couple of initiation sites. That's typical, that differ by a couple of bases. TATA box, and then a series of SP1 binding sites, repeated over and over, so you can get multiple proteins binding. And then the adenovirus early promoter, the E2 promoter, is different from the late promoter. Again, different protein are involved in regulating it. In particular, this E2F protein is important for the expression of this promoter, and we'll talk a little bit about that later. The point here is that every promoter is different because they have to be regulated in different ways. And they are regulated by proteins binding to specific sequences in the transcriptional control region, which is upstream of the initiation site on the promoter. So when I talk about regulatory sequences, this is what I mean, these sequences in DNA that bind proteins and have a role in regulating transcription. So this is a picture of an RNA polymerase II initiating transcription on a promoter. And I want to show this to you for two reasons. First, it's pretty complicated. There are a lot of proteins involved here. You see the RNA polymerase, the brown protein, is one of many. So by itself, it will transcribe, but it needs other proteins to be regulated and to do high-level transcription. And you can see the names of these, again, aren't very interesting. TF2D is the protein that binds the TATA box that I showed you right next to the transcription start site. And then there are lots of proteins called TF something, transcription factor something, and people were identifying these as column fractions and just named them TF22, TF23, DE, A, F, etc. So they're not very informative. But they all form a complex at the transcription start site, and they're all important for transcription. So that's the initiation complex, and this has formed, again, by binding of proteins to specific sequences around the promoter, and then the polymerase will start making mRNA. And again, you can regulate how much mRNA is made and when it's made by the levels of lots of these other proteins. The other point I want to make here on this slide is how these long distance sequences affect transcription. And so here's an example of an enhancer in orange. It can be up to 10,000 bases away. An enhancer will stimulate transcription, and it could also be a silencer that it silences transcription unless a condition arises where you need transcription, then you would take the silencer binding protein away. So here you can see that this enhancer works because cell proteins will bind to the enhancer, and this DNA will wrap around itself, and those cell proteins will interact with one of these transcription proteins and facilitate transcription in some way. So that is how the long distance works, via these binding proteins, sequence-specific DNA binding proteins. In this particular case, the interaction stimulates transcription. As I said, in other cases, the interaction can silence it by reducing the polymerase's ability to make mRNA. So let's take an overview of some of these proteins that regulate transcription. I've told you about some of the sequences that are involved already. These can be host, cellular, or virus proteins. We call them sequence-specific DNA binding proteins because they recognize very specific sequences around the promoter. So the TATA box sequence, which binds TF2D, is different from, say, an SP1 binding site. The protein will only bind a sequence that it recognizes, will not bind a different one. So we call those sequence-specific DNA binding proteins. There are also what we call viral co-activating molecules. These are virus-specific proteins that do not bind DNA, but they can modulate transcription. So they're often made by the virus to stimulate, say, transcription of viral genes using the host cell machinery. And they do not bind DNA directly, but they bind some of these other proteins that I've just talked about that bind around the promoter. And many of these work by modulating either the structure or the activity of nucleosomal templates. Now, you know that in eukaryotic cells, DNA in our nuclei are wrapped around histones to form nucleosomes. So you see the very common repeated nucleosome structure. This is just three of many, many. Part of the reason to do this is to compact the DNA, but it also allows regulation of transcription. Because when the DNA is wrapped very tightly around these nucleosomes, it can't be transcribed very well. It has to be chemically modified, either the DNA or the histone proteins, to loosen up the chromatin and allow transcription. And histone methylation, so methylation of the histone proteins is one way to regulate transcription. Acetylation of histones is another way. We're going to see an example of that later. Even methylation of the DNA can be involved as well. So the point here is that these viral co-activators that don't bind DNA, they can regulate transcription by, in fact, modifying whether the chromatin is tight or loose. Tight can't be transcribed, loose can be transcribed. And those co-activators can be brought to the template by binding not a specific sequence, but some of these other proteins that are bound to the promoter region. Now, this is what a typical sequence-specific DNA binding protein looked like. Called these sequence-specific transcriptional activators, or they're proteins that bind in the promoter region and stimulate transcription. They're typically modular. They have an N-terminal DNA binding domain, which is characterized by zinc fingers and basic helix-turn helix structures. There's a leucine zipper in the middle that allow these proteins to become dimers. They're often active as dimers. And at the C-terminus, an activation domain. This will bind another protein, recruit it to allow transcription to occur. And these are modular in the fact that is you can divide the molecules up and mix them among different proteins and get different specificities. So that's an example of the proteins that will bind DNA, specifically recruit other proteins to the DNA to establish a transcription complex. Our first question is, what is the first biosynthetic event that occurs in cells infected with double-stranded DNA viruses? Biosynthetic event: membrane fusion, transcription, DNA replication, protein synthesis, all of the above. The answer is transcription. Most of you got that. Biosynthetic event means you have to make a molecule. And so some of you picked a variety of the others. Membrane fusion wouldn't be a biosynthetic event. I understand why some of you would pick it, because before you can get the genome in the cell, you may have to fuse membranes, but it's not biosynthetic. DNA replication will not occur until you get at least one protein made. So to make that protein, you have to do transcription, which means RNA synthesis. And so you can't do protein synthesis before you make a messenger RNA. Here's an overview of how different viruses undergo transcription. Some viruses, the origin of the transcriptional components, which means the RNA polymerase and all those other proteins, is entirely host. And that includes retroviruses with simple genomes, polymerase, which are plant retroviruses, and circoviruses, which are single-stranded circular DNA genomes, very simple. I showed you in the second day some of the smallest genomes. These are among the smallest genomes encoding the fewest proteins. So these viruses are completely dependent on the host for the entire transcriptional machinery. And I have down here recognition of viral promoters. So those viruses where the DNA goes in the cell and it's transcribed by cellular proteins, the promoters have to be recognized by the cellular proteins. Otherwise, they would never get transcribed. Then we have increasing complexity here. The next level, these viruses are transcribed by mostly host proteins, but there's one additional viral protein that's employed. And there are two phages, bacteriophages, where there's a late protein that is actually a polymerase. This is, of course, in bacteria, where there is no nucleus separate from the cytosol. And so this bacteriophages have their own polymerase that only act late, because I think the reason is because they need to make a lot of structural protein to make new bacteriophages. Then we have, so that one viral protein for these phages is actually a RNA polymerase. Then we have another example of host plus one viral protein, where the viral protein actually regulates transcription of the viral genome, and that includes all of these viruses here: Adenoviruses, hepatitis B, the parvoviruses, single-stranded DNA, papilloma and polyomaviruses, and retroviruses with complex genome. Okay, so the transcription is mostly carried out by host cell components, but the viruses have a single protein that modulates transcription. We'll see an example of that in a bit. And then we have a more complex situation where, again, mostly host, but there's more than one viral protein that can participate in regulating transcription. And these are viruses with bigger genomes, adenoviruses and herpesviruses. And then finally, you have viruses where everything is viral. All the transcriptional components are viral. Nothing from the host is used. And those are the pox viruses and the mimiviruses, viruses that replicate in the cytoplasm. They have no access to the nuclear transcriptional machinery. So our cells, all of that is in the nucleus. The Pol2 and all the accessory proteins, it's all in the nucleus because that's where we do our transcription. So these viruses that are in the cytosol have to have all of their own machinery made. So you can see there is an increase in the left column from top to bottom in complexity in the number of in the number of viral proteins that are contributed from host only to viral only. And on the right, it kind of goes with genome size also. Smaller genomes are completely dependent on the host. The biggest genomes can encode everything they need to undergo transcription. Let's talk a little bit about how regulation of transcription fits into viral reproduction cycles. So first we'll look at some general principles here, and then I'll go through 3 examples of how regulation fits in with viral reproduction. Here, these are 2 examples of how transcription can be regulated by viral proteins. So here on the top, I have an example that I call cascade regulation. And the name says it all. One thing follows another. So here we have a viral DNA in blue, of course, and there are 2 genes shown encoded in it, gene X and gene Y, and each of them has their own promoter, shown by the red arrows. When this virus infects a cell, gene X's promoter is recognized by the cellular machinery. It gives rise to mRNAs that are produced by cellular proteins. That mRNA is translated to make protein X, which then is required for transcription of gene Y's promoter, or at gene Y's promoter. And that gives mRNAs that are needed for other things. This transcription is carried out by the cellular machinery, but the viral protein made, protein X, makes it possible. It wouldn't happen without viral protein X. So that's a cascade regulation where X is needed for transcription of gene Y. And you may wonder why you need to do such regulation. Well, you'll see in some detail in a moment, but in general, when the viruses infect the cell, they don't want to, let me back off and not say want. When viruses infect the cell, it's not efficient to make all the proteins at the same time. For example, you shouldn't make structural proteins in the very beginning because there's no nucleic acid to be packaged. That doesn't happen until later. So you might as well delay the structural proteins until you get nucleic acid synthesis. And that's one reason why you'd want to do regulation. And as you'll see, there can be some complex regulation going on in the bigger viruses. All right, so that's cascade regulation. At the bottom is what we call a positive autoregulatory loop. And it could also be negative, but this is an example of a positive one. We have a viral DNA comes into cells. First gene that is transcribed, gene X, it's transcribed by cellular proteins. It produces protein X, which then fits into the transcriptional complex and enhances transcription. And you get many more mRNAs made when you have viral protein X. So the promoter can clearly be recognized by cellular components, but it's not very efficiently recognized. But when you have that protein made, it stimulates transcription and you get more messages made. So that's a positive autoregulatory loop. In some cases, it can be turned around and be negative. So if you've made enough of this viral protein at a certain level, it can start to inhibit its own transcription. So this can be either positive or negative loops. So we're going to see examples of those now in 3 different viruses. We're going to talk about a simple virus, an intermediate virus, and a more complicated virus in terms of how transcription happens and how it's regulated. And so the first one is a polyomavirus. Again, these are smallish, double-stranded circular DNA genomes. They're built with icosahedral symmetry, no membrane. And the model organism, or the model virus, is SV40, which people have worked on for years to study transcription and DNA replication and tumor formation as well, as we'll see in a couple of weeks. The genome is a double-stranded DNA circle, 5 to 8 kb, 5,000 to 8,000 bases in length, not very big. Similar viruses to papillomaviruses. Human papillomaviruses cause warts or cancers even. And the transcriptional program is regulated. So even in this simple virus, there's an early and a late phase of transcription. And on the bottom is shown the DNA template. The red box is what we call an ORI, or an origin of replication. We'll talk about this next time. It's where DNA synthesis initiates. It also happens to be where the transcriptional promoters are located, and they go in opposite directions. There's a promoter for early mRNA synthesis, and there's a promoter for late mRNA synthesis. And they both start around the origin. So when these viruses infect cells, the DNA goes to the nucleus, and the first thing that happens is this early promoter is recognized by the host cell transcriptional machinery. Don't need any other viral proteins for that promoter to work, and you get early mRNAs made. And on the top line is this timeline of the transcriptional program shown as a line. So this, if you could think of as time after infection. So on the left is early after infection, when the DNA is first in the nucleus. You make the early mRNAs, and those encode, among other things, a really amazing and important protein called large T antigen, which we will see multiple times in the next few weeks. LT, large T antigen. Large T antigen is required for DNA synthesis. And so DNA synthesis is delayed until there's enough T antigen made. And when there is enough DNA made, that leads to the expression of the late genes, the L genes shown at the right here. And those are transcribed from the opposite direction from the ORI. That purple bar signifies the onset of DNA synthesis. So you can't make DNA until you have enough large T. So large T stimulates DNA replication, as you'll see next time. And when DNA replication occurs, it turns on the late promoter, which up until that time has been shut off. And that's why we say DNA synthesis has an anti-repression role. It allows synthesis of the late genes. The late genes encode structural proteins. They are made late because you don't have enough DNA to encapsulate until late in the infection. So that's why we have this two-phase transcriptional program. First, to make T antigen, which will allow DNA synthesis, and then once DNA synthesis begins, we can then make structural proteins. So it's a relatively simple transcriptional pattern, and you can see that there are cascade elements, right, where you need large T in order to get DNA replication. Large T also helps the activity of the late promoters as well. So let's put this in the context of a cell. Here is how the late promoter is turned on by DNA synthesis. So first let's look at this graph on the lower left. So on the bottom is time after infection. On the x-axis, on the y-axis is just relative concentration. And so the first thing we'll look at is in blue, viral genomes. So early in infection, there's not many viral genomes, but then they start to replicate as we move into the late phase. And then we can look at late RNA in green. There's no late RNA early. That's why it's called a late RNA. But it comes up only when viral DNA starts to replicate. And the key player here is a cell protein called the initiator binding protein. And its concentration is shown here as a yellow line. And you can see it doesn't change throughout infection. So here's what happens. When the virus first infects a cell, let's go to the right-hand side now. Here's the viral genome. And that red arrow is the late promoter. When that genome comes into the cell, it's coated with initiator binding proteins. There are binding sites for those on the viral DNA. So as soon as it comes in the cell, those binding sites are all occupied by initiator binding protein, and they repress the late promoter. And this is evolved to be this way. The viral genome has evolved to have these sites that bind IPP, which is constant throughout the reproduction cycle in the cell, represses late transcription. So at this point, we're only getting early mRNA synthesis, including T antigen. When you get enough T antigen made, by translation of the messenger RNA, of course, then DNA starts to replicate, and you start to make more and more copies of the genome. Because the concentration of IBP remains the same, that effectively titrates the repressor out. So you get the same number of IBPs binding to more and more DNA molecules. There are not enough IBPs to occupy all the binding sites. So eventually you're in a situation where you have way more DNAs than IBP molecules, and that allows the late promoter to become active because IBP can no longer bind to the DNA and repress the promoter. So you can see now you're getting late transcription, and those late messenger RNAs encode the structural proteins because now we have enough viral DNA, we can start to encapsulate it. So it's a very nice mechanism for keeping the promoter shut off early in infection and having it turn on later, which is controlled by DNA replication. So that's the late promoter of the virus. So this is a view of the whole thing in the cell cycle. I haven't shown you this yet. We have our virus binding to cell surface receptors. It's taken in by clathrin-mediated endocytosis, which delivers it to the endoplasmic reticulum. The virus deposits its DNA into the nucleus. And this virus is unusual in that in the virus particle, the DNA is already wrapped around nucleosomes. Most viruses, it's naked DNA or bound to a few proteins. But here it's already wrapped around nucleosomes. And that's why in these images, if you look closely enough, or if you blow up the PDF big enough, you can see there are nucleosomes in the middle of the particle. So that DNA enters the nucleus already wrapped around nucleosomes. It is ready for transcription. It's double-stranded, fully double-stranded. And so that goes in the nucleus, and the first thing that happens is the production of early mRNAs, which encode large T. So those mRNAs have to be made in the nucleus, of course. They go out into the cytosol. They're translated. Large T then goes back into the nucleus, and it can start stimulating DNA replication when it gets to a high enough concentration. And so now you see the DNA is being replicated, again, wrapped around nucleosomes. And once enough DNA is made, that turns on the late promoter. So now you have the late phase shown here by this dotted line. There actually is no dotted line in the infected cell. You know that, right? It's just for us to look at. The late phase is occurring where we have DNA replication and transcription of late genes, which encode all the structural VPs, viral proteins here, to build the capsid. So again, those have to be translated in the cytoplasm, shipped back into the nucleus to assemble around the DNA to make new virus particles. So that's the function of the early and late phases, to delay synthesis of structural proteins until the DNA has been replicated. And it's achieved by transcriptional regulation, by having a T antigen first that turns on the genes that are needed for DNA replication. All right, so that's a simple example. Let's get a little more complicated. Adenovirus, so SV40, about a 5 kb genome, now 35,000 base pairs of linear double-stranded DNA for adenovirus. And here it's a little more complicated, but the same diagram at the top, a timeline of transcriptional regulation. Here we need 3 viral proteins and DNA synthesis, very much like SV40, to govern the phase transitions. The genome comes in, it is immediately transcribed at an immediate early promoter. So here we're getting a little fancy, not just early and late, but we have immediate early, early, and late. And the immediate early transcription gives rise to this lovely purple protein called E1A, which is needed for activation of the early promoter. The early promoters will not work without E1A. And we'll see why in a moment that happens. The early genes include genes needed for DNA synthesis. And they include E2, another protein that is needed to stimulate DNA synthesis. And once DNA synthesis happens, the late genes can be turned on in a similar mechanism as we just discussed for SV40. These include structural genes, and there are a set of proteins here shown as 4A2. Those are made as late genes, and they also stimulate the production of late genes themselves. So they are kind of we talked about this mechanism a few slides ago, where a gene will stimulate its own transcription. They're doing that for the late gene. So E1A is needed for all these early transcriptions to occur. And E2 is required for DNA synthesis. DNA synthesis allows entry into the late phase, and then the late phase gives rise to the mRNAs that encode all the structural proteins. So let's take a look at that in more detail. So what's this E1A protein, this little purple protein? What is it doing? So it turns out there are promoters on the adenovirus genome. So if you look at the blue DNA on the right side, imagine that's a part of the adenovirus genome, and there is a promoter, it's transcription initiation site, and upstream is a binding site for transcription factor called E2F. And this is needed to get transcription at this site. So adenovirus promoters require, the early promoters require this cellular transcription protein called E2F to bind to that site, otherwise they don't work. And so that's why E1A is made, so that it can get these promoters to work. So here on the upper left is the E2F transcription protein. It is bound to another protein called DP1, and normally is bound to a third protein called RB, retinoblastoma protein, which we'll revisit many times in this course. And if these three proteins are bound together and they interact with the E2F site, it doesn't allow transcription to occur. The presence of RB represses transcription. The reason is because RB recruits a protein called a histone deacetylase, or HDAC. HDACs will bind to RB. They will deacetylate the histones around which the DNA is wrapped. That tightens them up and it shuts off transcription. Now, why this happens, we'll get to in a couple of lectures, why RB would do this. For now, you realize that adenovirus has to somehow overcome this inhibition caused by the RB protein. Because as the virus comes into the cell, these E2F RBs will bind to its promoters and silence the genome, would never get anywhere. That's why the virus encodes an E1A protein, which is made by an immediate early promoter that doesn't require E2F. That promoter will fire as soon as the viral DNA gets into cells. E1A is made, and what does E1A do? It binds RB. It binds RB and pulls it off of E2F. And that frees up E2F to be able to activate transcription at the early promoters of the virus. All right? So that E1A protein is essential for getting all the subsequent genes expressed because it gets rid of RB. So this E2F protein, of course, is a cellular protein. It's not a viral protein, and it's regulating cellular genes. So keep that in mind because we're going to come back to that when we talk about how viruses cause cancer. Anyway, the E1A protein's function is to bind RB, pull it off of the E2F, and allow E2F to act as a promoter of transcription. So that's the adenovirus genome, 35 kilobases of a double-stranded genome. And these are the mRNA transcripts that are made. Here, the immediate early transcripts on the left, E1A, E1B. These are proteins essential for early activities, as I've just told you. There are some other early transcripts scattered around. Here's another one here, E4, E3. You can see transcription occurs from both strands of the genome. You get transcription from the top strand and the bottom strand. So those are early genes. And then we have, let's see, sorry, these are early transcripts, the E2 and the E3 and the E4. They're not immediate early, they're early. And then we have the late transcripts. We have this one here, the major late transcript, which goes almost the entire genome. As you can see, it encodes many structural proteins. We'll see how that works in a moment. Oh, I forgot the E2 transcript. It's another early transcript, which encodes the DNA binding protein and the DNA polymerase. So this is needed to get the genome to replicate. So E1A, this little guy, E1A, is essential for getting all these E transcripts made. And this one is very important for getting the genome replicated. So here's an overview of the infectious cycle. Again, this is a little more complicated than SV40. We now have 3 phases when the viral DNA gets in the nucleus. So the virus is taken in by endocytosis, pH drops, particle punches out of the endosome, docks onto the nuclear pore, the DNA goes in the nucleus, it's double-stranded, ready for transcription, and the E1b promoter is recognized, the immediate early promoter is recognized by cellular proteins. You make immediate early transcripts. They go in the cytosol, they make E1a and E1b. Those proteins go back in the nucleus, they activate the early promoters, so now you have a second round of transcription, early transcription, which includes the DNA polymerase and other enzymes involved in DNA synthesis. They, of course, have to go back in the nucleus and assist in the replication of the genome. And finally, when the DNA replicates, that activates the late genes by anti-repression, as we talked for SV40. And those genes encode the structural proteins, which again have to get shipped back into the nucleus because that's where they package the newly made DNA, and eventually the newly made virus particles come out. Okay, so that is, I think it's brilliant because the immediate early promoter will work, but the rest do not work unless you make E1a and antagonize RB. The last example is Herpesvirus, where again we have immediate, early, and late phases, but a little more complexity. Here, what I find really interesting is that the immediate early promoter of the virus, so the analog of the promoter for adeno that would make E1b and E1a, that promoter of herpes simplex does not work without a viral protein being present. So how do you deal with this? You got viral DNA coming in the nucleus. You can't transcribe that immediate early promoter to get the mRNA synthesis going. So what do? Well, you package the protein in the particle, right? It's like negative strand RNA viruses. They package a polymerase because that's what they need to make mRNAs. Here, the viral protein is called VP16, and it's packaged in the space between the herpes virus capsid and the membrane. So these are viruses with an icosahedral nucleocapsid. They're surrounded by an envelope. The space in between is full of proteins, and that's what all those little dots are in the image. And one of those proteins is VP16, and that's needed to start the immediate early promoter. And that in turn turns on early proteins, which include proteins you need to replicate the genome. And once the genome is replicated, you then turn on the late genes, which are needed for making structural components. So it's similar to adeno, except this interesting complexity where you need to bring in a viral protein to get the whole thing started to begin with. So a great example of regulation. Again, the idea is keeping the late genes after you got DNA synthesis occurring. And so here's the replication cycle of a herpes virus. Just like adenovirus, 3 phases: immediate, early, and late. So we have virus binding to the cell surface, and these viruses fuse at the plasma membrane, so they are low pH independent. And the nucleocapsid comes out, moves down microtubules, docks on the nuclear pore. And remember, there's one portal in the herpes virus capsid that docks onto the pore. The DNA comes out into the nucleus. That DNA is dead in the water. It can't be transcribed unless VP16 comes in with it. So here you see, as the virus is uncoating at the plasma membrane, all these little dots coming out, those are the viral proteins that are between the capsid and the membrane. And among those is VP16, which has a nuclear import signal on it. So it gets in the nucleus, there it is, and that will allow the immediate early promoter to function. So again, the cell transcriptional machinery doesn't recognize the immediate early promoter of this virus. It needs VP16 in there to do that. And then you get the immediate early mRNA made, it gets exported. The proteins are made, put back in the nucleus. Now you can have early gene expression, which will include a DNA polymerase. And that DNA polymerase will replicate the genome. And finally, when the replication occurs, you get late proteins made, which are the structural proteins that give you assembly of the capsid, and eventually that buds out. We'll talk about assembly of all these viruses separately, because that's a very interesting topic as well. So again, tightly regulated to make sure you make late proteins and DNA at the right time. Our next question is, adenovirus E1a protein stimulating the expression of adeno E2, which then stimulates the expression of 4A and L4 protein, is an example of a negative autoregulatory loop, repression of gene expression, cascade regulation, or dimerization. So the answer is cascade regulation, which most of you got. A few of you said repression, but this is just, in turn stimulation, so I don't think repression would be the right word for that. All right, so the other aspect of transcription is getting a mature messenger RNA. So let's talk about that. First is the addition of a cap, which happens very early in transcription. You can see the final mRNA has that cap left on it. This is a cap on the right, which I haven't shown you before. So that base 1 is the first base in the viral RNA, then base 2, et cetera. And a cap is the structure here. It is another base, just a normal base, typically a G, guanine, and it's linked to the first base, but it's a 5' linkage, not 5' 3', which usually happens in the rest of the nucleic acid. All right? So it's 5', 5', and there are 3 phosphates left, not 1, as you find in the remainder of the chain. So it's unusual. It's called a cap, and it's essential for translation, for mRNA stability, and many other activities. This cap is added shortly after transcription begins. And here's a depiction of that. That's our DNA template with the initiation site in red. The polymerase has bound. It's starting to make some mRNA, as you can see in green. The polymerase gets phosphorylated in its C-terminal domain, and that allows the capping enzyme to join and add the cap to the 5' end. And so you don't cap before you make the first nucleotide of the mRNA. You make a little bit of mRNA and then you cap it, and I think the reason is to make sure that this is going to be an mRNA, that the polymerase is not falling off early on, which often happens. And if you're making an mRNA for 20 or 30 or 50 bases, then the cap will get put on. At the other end, you have to put a poly-A tail, and I just want to briefly mention how this works. So there's, in the nucleus, this is where this happens. This happens after transcription ends at the 3' end. So we have, there's our pre-mRNA that's made. There is a poly-A addition site in the mRNA, which is synthesized as part of the primary transcript. And then the polymerase keeps going to make a longer part of the mRNA. But there's no poly-A included in this mRNA. Rather, there's a poly-A addition site at which a cellular enzyme will come and cleave, and a series of reactions occur. But basically, the cleavage site is going to be the 3' end of the message. There's a poly-A polymerase reaction that occurs, adding A's onto the end until you get about 200 A's added. So there is a recognition of a sequence, cleavage. The 3' end is then polyadenylated by enzymes. And this is independent of having a template. You don't have a poly-U template like we have on some viruses. This is a non-templated addition of poly-A. So I want to compare this polyadenylation among the different viruses we've talked about. And so the DNA viruses, they're polyadenylated by a cellular enzyme that cleaves the RNA and then polyadenylates it. Whereas RNA viruses that we talked about last time, the polyadenylation occurs during mRNA synthesis. And so for influenza and VSV, we saw how you get reiterative copying of a short stretch of U as the polymerase is stuttering and it makes long poly-A sequences. Or for polioviruses, there's actually a long U stretch in the template that's copied to give poly-A. So very different mechanisms of polyadenylation in RNA viruses, where again, it happens during mRNA synthesis, whereas for these DNA viruses, it happens after transcription is finished. Now, an important aspect of transcription, of course, is splicing, which was discovered in adenovirus infected cells. And the observation was that some of the adenovirus mRNAs seem to be made in a longer form in the nucleus. They were way longer than the final mRNA. And the investigators who were studying this couldn't figure out what was going on. So someone suggested that they look at these RNAs under the electron microscope. And so they took the mRNA for one of the proteins called the hexon. It's a structural protein of the capsid. That's shown here in the middle in the cartoon in green. And they hybridized that to adenovirus DNA, single-stranded adenovirus DNA, which they made by denaturing the genome. And when they looked at this under the electron microscope, they saw the pattern shown on the upper right here. That's an exact tracing of the original electron micrograph. And I've simplified it here in the middle. They saw basically that there were big loops of the adeno-DNA when it was hybridized to the message. The suggestion being that there was extra sequence in the DNA template that wasn't found in the final message. And we now know that these loops are introns that are removed from the precursor to mRNA by splicing. And so this hexon mRNA, for example, is transcribed as a long precursor, and the blue, the orange, and the red are the loops that are coming out. Those are the introns that are removed to give the final message. This was an important discovery, and 2 investigators got the Nobel Prize in 1993 for discovering this. So it was discovered in virus-infected cells, but pretty soon afterwards it was found throughout eukaryotic cells and in many viruses as well. And again, the idea is that you have a pre-mRNA which has intervening sequences or introns that need to be removed by the process called splicing. And people began to sequence many of these pre-mRNAs, and this interesting consensus emerged. So at the top is a consensus sequence of thousands and thousands of mRNAs. So on the left you have the first exon. So exons are the parts of the message that end up in the messenger RNA. And then the red is the intron, and at the right is exon number 2. So in splicing, this red intron would be removed and the 2 exons would be joined together. And looking at the sequence, you could see that there were very high homologies. These are frequency of occurrence at the bottom here when you're looking at thousands of different mRNAs. Highly conserved sequences at the ends. And in the middle, this particular A, 100% conserved in the middle of this intron. And work in many laboratories showed that these conserved sequences are part of the reactions that lead to splicing. So what happens here to remove this intron, look at panel B on the left, you have our 2 exons, the intron is in pink. That conserved A, that very much conserved A, its 2' hydroxyl attacks the phosphodiester bond at the intron exon border. It cleaves it. It's called the first transesterification reaction. So now you have a bond between that 2' hydroxyl and the phosphate. That exposes a hydroxyl at the 3' end of the first exon. That hydroxyl attacks the second phosphate. The exons are joined together, and you have an excised lariat intron released. And these can be seen in cells. It looks like a lariat, right? The ropey things, and that's how splicing works. Now you may wonder how can this happen with things not floating away and being lost? Because it happens on a structure called the spliceosome. And a spliceosome is made up of nothing more than small nuclear RNPs, ribonucleoproteins, which are shown here as colored RNAs like U1 and U2, and behind them are the spheres. These are proteins. So here we have our pre-mRNA with an intron. These short RNAs that are made in the nucleus base pair to conserve sequences in the introns of these pre-messenger RNAs. And that's how they're held in place for these transesterification reactions to occur. So you can see base pairing between the U1 and the first exon boundary. There's base pairing between U2 and the conserved A region there. And multiple of these snRNP proteins come in to carry out all of those reactions, which will eventually lead to release of the mature mRNA. And you see the intron is remaining associated with the small nuclear RNP particles. This is called a spliceosome, this structure on which splicing occurs, and it's catalyzed by the RNAs. The RNAs are what are catalyzing those transesterification reactions that lead to splicing. And so we call it a ribozyme, and we think it's a relic of the RNA world when RNA had both informational and catalytic activity. So the point here is that all the splicing occurs on this RNA protein complex. And splicing gives you flexibility in gene expressing. You can imagine if you have an mRNA, say, with 2 introns, shown on the left. You could splice both of them out and get 3 exons, 1, 2, 3. That's called constitutive splicing when we remove all the introns. But you don't have to remove all the introns. There are signs at the ends of the exons. There are proteins that bind them. They can regulate whether an intron is removed or not. So you could have alternative splicing. You could get not just 1, 2, 3 exons, but skipping number 2. So you splice from the 3' end of exon 1 to the 5' end of exon 2. You can get alternative splice sites where you have different acceptors utilized, and that can be alternative 5' splice sites or alternative 3' splice sites. So in other words, having introns lets you make a lot of different messenger RNAs from the same primary transcript. And this, of course, gives you flexibility in gene expression, particularly in viruses with small genomes. So here's an example of how amazing alternative splicing can be. Here's our adenovirus genome at the top, and that is the major late promoter, 16,500 bases from the left end, and it gives rise to a huge mRNA, you can see there, and that mRNA can be spliced to form dozens of individual RNAs for different proteins. The way that happens is by a combination of alternative splicing and alternative polyadenylation. So this major late transcript contains 5 possible polyadenylation sites, L1, 2, 3, 4, 5. And so you could get initially these collection of nested transcripts by polyadenylation at each of those different sites, but you can see those have introns in them. And they can be spliced alternatively to give you all sorts of different proteins, as you can see on the left and on the bottom. And that's how you get so many different proteins from a single messenger RNAs. So splicing gives you flexibility, and of course viral proteins can regulate alternative splicing. As I said earlier, I didn't show you how this works, but there are signals at the introns and the exon junctions that regulate whether a junction is going to be spliced or not, so viral proteins can manipulate the kinds of mRNAs that are eventually made. Next question is, which statement about polyadenylation of DNA virus mRNAs is correct? It always occurs in the cytoplasm. It occurs after cleavage of pre-mRNA. Poly-A is added at the 5' end of pre-mRNA, and it is specified by a stretch of U residues in the template. And most of you got it occurs after cleavage of pre-mRNA. That's correct. It doesn't occur in the cytoplasm. That's a nuclear activity. It's not added at the 5' end. It's added at the 3' end. And it is not specified by U's in the templates. That's an important point I wanted to make. It's added post-transcriptionally. All right, 2 quick things before we finish. First, splicing designates mRNAs for export. In general, if mRNA is going to be exported, it has to be spliced. And that's because all the proteins that are attached to the RNA on the spliceosome, which are shown here, some of them remain on the mature message. Those are recognized by the nuclear export machinery. So here, NFX1 is a nuclear export protein. It recognizes the proteins that have been put on the spliced mRNA for the splicing process. This is what causes mRNAs to be exported. In other words, splicing marks mRNAs for export. The problem is that some mRNAs, some viral mRNAs are exported without splicing. For example, retroviral RNAs, as we'll see, some of them have to be exported without removing the splice so that you can get genomes made. And that is done because the viral genome has a sequence at the 3' end called a CTE, a constitutive transport element, that actually binds elements, proteins of the nuclear export pathway, and that's what gets the unspliced RNA out of the nucleus. So normally, again, you would need to be spliced to have the nuclear export machinery recognize the mRNA, but here the genome of the virus has a signal that kind of co-opts the export machinery. So that's one kind of retrovirus that does that. The other is HIV, where the full-length genome eventually has to be exported unspliced so it can be packaged into virus particles. This genome is made, this mRNA is eventually spliced to form a very short message that's exported. It's completely spliced. It makes a protein called Rev. Rev goes back into the nucleus and binds the Rev responsive element on the genome, and that is recognized by the nuclear export machinery and gets unspliced or limitedly spliced messages out of the nucleus. And so that's how you can get genomes out. So in other words, these viral genomes have found ways to get around the requirement for splicing by having either sequences in the genome that recognize the nuclear export machinery or that attract some of their protein. Today we have talked a little bit about splicing. I want you to remember that it creates different mRNAs and proteins. You can expand the coding information of a small genome, but also splicing can regulate gene expression depending on how splicing is done at different times in infection. And these, again, are the genomes we've talked about. The genomes ready for transcription, double-stranded, and the ones that have to be repaired before. Next time, we'll take a look at how those DNA genomes are replicated in the late phase of infection.